How do you solve a crime with just a single hair? Or how do you diagnose a genetic disease before it even starts? Well, the answer to all of that, believe it or not, lies in this really brilliant technique for sorting the invisible. It's called gel electrophoresis.
So let's start with a big question. Think about it. DNA, proteins, all the molecules that literally make us who we are, they're completely invisible. You can't see them. So if you've got this microscopic soup of all this stuff, how on earth do you even begin to organize it? How do you pick out the pieces that you need to look at?
Well, the answer is kind of genius. You make them compete. You set up this tiny molecular racetrack, and you make all those little fragments run a race that's powered by electricity. And that right there is the secret behind this whole process.
Okay, so this whole idea of a molecular racetrack, it sounds pretty cool, right? But what does that actually mean when you're in a lab? Let's break down what gel electrophoresis really is. All right, here's the official definition. It's a technique that scientists use to separate things like DNA fragments. And it all comes down to just two key properties, their size and their electrical charge. That's it. Everything else we're about to talk about flows from those two simple ideas.
So this gives us the two simple rules of the race. First up, size. Just imagine you're trying to run through a really thick forest. If you're small and nimble, you can zip right through the trees, no problem. But if you're bigger, you're going to get snagged, you're going to move slower. It's the exact same idea for DNA fragments moving through the gel. The smaller they are, the faster and further they go. Simple. Second rule, charge. DNA molecules naturally have a negative charge. And, from basic physics, opposites attract. So when you flip on an electric field, that negative DNA gets pulled straight toward the positive end.
Okay, so we know the rules of the race now, right? Small and fast, and pulled towards the positive pole. So let's head to the starting line and see how this race actually gets run.
And while it might sound like something out of a science fiction movie, the actual process is, well, it's beautifully simple. It really all boils down to three key moments. First, you load the runners. Then, you fire the starting gun. And finally, you just watch the race unfold. Let's take a closer look at each of these steps.
So step one is all about getting our runners to the starting line. The gel itself is kind of like a slab of jello, and it has these little pockets at one end called wells. Scientists use a super precise tool, a pipette, to carefully load their DNA samples into these wells. Each little lane you see can be a totally different sample from a different source. And now for the moment of truth. An electric current is passed through the whole gel. The end where the DNA is gets a negative charge, and the far end gets a positive charge. This is basically the starting gun. That negatively charged DNA is instantly repelled from the negative side and pulled, like a magnet, toward the positive side. The race is on.
And this is the result. Over a little bit of time, all those fragments start to sort themselves out. You can actually see these bands forming in the gel. The smaller, faster fragments, they just race ahead, forming bands way down at the bottom. Meanwhile, the bigger, slower fragments, they get left behind, forming bands closer to the top where they started. What you're left with is this beautiful, clear pattern of DNA, sorted perfectly by size.
Now, watching this race is fascinating, for sure, but the real power, the real magic, comes from what we can actually do with these results. So let's see how this little lab technique makes a huge impact in the real world. So we've got this incredible tool for sorting the invisible. But so what? Why does it matter? What can you actually do with a neatly sorted pattern of DNA fragments? Turns out, you can do a whole lot. The applications here are just huge. In forensics, this is the very foundation of DNA fingerprinting, the technique that has solved countless crimes and even exonerated innocent people. In medicine, it's used for genetic analysis to find inherited diseases, giving families absolutely crucial information about their health. Researchers use it to hunt down the genes linked to things like cancer, which helps pave the way for new treatments. It's really the unsung hero of the modern biology lab.
When you boil it all down, that's what this is all about. Gel electrophoresis gives us a way to see the unseeable. It takes that abstract, invisible code of life, our DNA, and it turns it into a clear, visual pattern that we can actually read and understand. It makes the code of life real. And that really just leaves us with one final thought. We now have this incredibly powerful way to read and sort the fundamental instructions for every living thing on this planet. So the only question left is, as we get better and faster at doing this, what incredible secrets are we gonna unlock next? What will we learn about our health, our history, and our world?